So thank you again, Srinivas, for being here today. I took a few notes to avoid to make some mistakes in my presentation. So you are working at Georgia Institute of Technology, where you are both professor and director of the Institute for Data Engineering and Science. And you have been working on an impressive different topics in bioinformatics, in the error correction of reads, of short reads, long reads, mapping directly nanopore signals extremely fast. And recently you have been working more on genomics, in parts on what we have seen last week, 2 weeks ago with Hangley. You are working with him on this topic of the wave front algorithm for mapping on graphs, and you are working also on haplotype aware variant detection selection. So I think you are going to present us some of these works today and mainly what you are doing on genome graphs. So thank you very much again for being here, and the floor is yours now.
Okay, thank you, Pierre, for the kind introduction. I hope everyone can see the slides right now.
Yeah, that's perfect here.
Yes.
Okay. Thank you. So the way it's working out on my computer is that I'm in a presentation mode, which means that I'm only seeing the slides. I'm not seeing any of you. So if you would like to ask a question, please feel free to interrupt, and then I will be able to hear it. But I'm not able to see you or see the chat. So please speak up if you want to stop me for any reason. Okay. So first of all, thank you very much for inviting me to give a presentation to this rather sizable group that is focusing on working on genome graphs. I hold your work in high esteem. I know these European projects that you're involved in pangenomics, and my group tries to follow up and read your publications and so forth. So it's a real pleasure for me to have an opportunity to also present our work in this group. So thank you for inviting Hank last meeting and me now, so we could share our ideas and what we've been doing to work in this area. So this is one of the 3 research focus areas in my group. So there are a number of people involved in this work. So I want to start by acknowledging them. My current and former members of my research group, Daniel Gibney, a very bright postdoc in my group. He's here. He's able to join us in the meeting. So Daniel, when it is appropriate, feel free to take questions as well. PhD students, current and former, Chirag Jain, Neda Tawakoli, and Havan Jang. So the work I'm presenting actually reflects the work of all of these people in my group, as well as some external collaborators, including Hank, who you heard last time from Harvard, Sanchit Mishra from Intel Corporation, a former research scientist in my group, but now an associate professor at NCSU, Sharma, and also Alex from Germany. So this is really group work. And the work that I'm presenting today it really covers the material that we have in several publications. And for that reason, I won't be able to go into detail of every algorithm and every proof. But what I will try to do is to give you a high-level overview of the work, lay out the rationale and motivation behind it, the impact if you use it in practice, and then maybe go into details of algorithms to some extent in some areas, so that you get a feel of how this whole thing works. So please feel free to stop me at any time to ask questions, so that this doesn't end up being a monologue. Okay, so I may quickly go over the introduction slides, because your group, everyone here already works on this problem, so I don't need to introduce this problem, right? So a couple of decades ago, we just had a single reference. And since then, we are sequencing more and more genomes, particularly human genomes, but this is happening with other species also, not only mammals, but also plants and so forth. So when you have a population of genomes sequenced, it's natural to try to move beyond the single reference and then try to use graphs to index multiple genomes simultaneously in a compact fashion, and then reinvent the bioinformatics that we know, which is based on sequence to sequence, to the era of sequence to graphs, right? So this is what your group is prioritizing and working on, and this is what we are working on. So there's no need to convince you of the importance of this problem. There are, obviously a number of papers by leading researchers, in fact, several of them here are European, talking about the reason why we should be using genome graphs and how that will beneficially impact the applications and so forth. So, briefly, the advantages of using genome graphs are we are able to capture the genetic diversity in the population and not treat everybody's genome as identical. It also reduces the reference bias in genome mapping. So if you're trying to sequence a new individual of the species and you generate reads from a high throughput sequencing instrumentation, if you map it to a single reference, there is a reference bias. And if you map it to multiple references simultaneously, then you would expect that your mapping would be better. And there are many papers that talk about using genome graphs in applications ranging from variant calling to genome assembly to read error correction to RNA-seq data analysis and more. So the challenges, as I mentioned already, is that we have decades of bioinformatics research that is in the area of sequence to sequence alignments. Now we need to sort of reinvent the wheel, so to speak, in the era of sequence to graphs. And maybe there are even some applications where we need to think of graphs to graphs, right? Like maybe merging variation graphs and things like that. So this talk will focus on answering two key questions. So one is that suppose that you have a large number of genomes sequenced and the variants are cataloged, like in the 1,000 Genome Project, etc. How do you take these variants and construct the variation graphs, right? So that is the first question. The second question is that the killer application here is sequence to graph alignments, right? So how do you take a read from a next generation sequencing machine that may correspond to a different individual, perhaps within the same species population, and how do you align that read to the graph that you have? So these two questions are obviously interrelated in the sense that you're constructing variation graphs so that you can perform alignment on them. So the answer to one question may be influenced by how we answer the other question. So my talk itself will be divided roughly into two equal segments. I'll spend the first half an hour on our ideas about constructing variation graphs, and then the second half an hour about performing sequence to graph alignments, and maybe a natural stopping point to take some questions after I finish the first part. So variant selection. So here is an example. So consider three haplotypes here, R1, R2, and R3. And I'm showing you what we call a complete variation graph. This is a graph that captures all the variations that you see in all of these haplotypes. So for example, you will see that in this position that I'm highlighting, there is a G in R1, A in R2, and C in R3. So by default, we take R1 to be the default reference genome, also a backbone in this graph, in the sense that we build this graph by first crafting a path with one character label per edge that exactly spells out R1. There's nothing sacrosanct about R1. We can as easily choose any of these references to be the backbone, but just for convenience, I'll always choose R1. Also, at a particular position, so here we have a SNP or a single nucleotide polymorphism, right? Sorry, I'm racing here. So in this position, you have a G in one haplotype, A in another, and C in another. So you can show that SNP like this in the graph, where between two consecutive vertices, then you have three edges, multiple edges, and with different labels. So when we try to remove variants, if you were able to do that, we always keep the variant in R1 as the default. And again, there's nothing special about it. We can as easily choose any of the other bases, and all the theory and algorithms would remain true. So you don't need to really worry about it. But just as a convention, I will give R1 a special status, call that the backbone, and so forth. So as you can see, if there is a SNP, we take two consecutive vertices like that and have multiple edges. And if you have a deletion, so for example, R3 is missing ACA, then essentially you're connecting two vertices that are some distance apart by an edge labeled epsilon, okay? And if you have an insertion like this, for example, GTC in reference R2, then you have a loop essentially right here at this vertex, where you can take this path GTC and come back to it essentially. And when you do that, you are following this insertion variant. So this is what insertions look like. Epsilon is what deletions look like, and so forth. And SNPs will look like this. And so we really have a graph where the vertices can be laid out along the coordinate axis. And therefore, we can even talk about the coordinate of a vertex in this complete variation graph and so forth. In this model. So the question is that why not just use the complete variation graphs, because they're not that hard to build. You have your sequences, all the variations, so you can just walk through and build this graph. And so why not just do that and use them? So the answer is that when we are trying to do read to graph mapping, the read is mapping to the sequence given by a path in that graph. And what can happen is that because this is a graph, you may start by mapping to some portion from R1 and then jump to R2 and then jump to R3. So this is what we call recombinant paths. So the paths to which that your mapping reads may exist in the graph, but may not exist in any of the input haplotypes that we have observed so far. So these recombinant paths may or may not be sequences that you would see if you sequence every individual in the population, and therefore they could be biologically incorrect and could lead you to wrong conclusions. And because the number of paths is combinatorially explosive, the more variants you add, the more these recombinant paths, and then they will dominate. And then your mapping results, the accuracy of mapping may actually go down. And many empirical studies demonstrate that as you start adding variants, you see benefit. But then if you try to add beyond a certain threshold, you actually see diminishing returns, decrease in accuracy at extreme cases. The other problem is that
Srinivas.
Yeah, go ahead.
Solon, a very quick question. Is there any reason why you choose this, for instance, this cycle like GTC, whereas, for instance, these deletions, I see that in other parts you use epsilon transitions. So is it just because, one could make this to be a directed acyclic graph, right? Why do you use cycles in particular?
Yeah, so I agree that there are multiple different models for looking at genomes. And I think the directed acyclic graph model simplifies algorithms, but is not really the perfect model in every case, right? For example, if you have repeats in a genome, it makes sense to go over cycles and so forth. So when we develop the theory, which you will see later, we will try to address general graphs when we can, but we also work on restricted cases like DAGs and so forth.
Thank you. Yes, makes sense. Thank you.
Okay. And so when we talk about the complexity of mapping algorithms, and again, this is the second part of the talk, and there you see that many problems there are NP hard, and therefore the larger the graph size is, the more time that they're going to take. And because the goal of building these variation graphs is to try to do efficient read mapping from sequence to graph, we also want to keep the graph size reduced if we could for that purpose. So that's the motivation for selecting a subset of variants and not really cataloging all the variants in the variation graph. So the problem that we are interested in is that given a complete variation graph and a set of haplotypes as input, we would like to construct a reduced variation graph which only catalogs some of these variants. So as you can see here, we have 3 SNPs here, and we only kept 2, and for some reason we discarded 1, right? And we kept this deletion, we threw away this insertion, we threw away this deletion, and we kept this insertion, and so forth. So you choose to keep some, retain some, and you choose to throw away some. And the question is that what is the right way to do this? How do you come up with the right reduced variation graph? Okay, so before our work, the way this problem was solved is a pretty ad hoc basis, right? So here is an example of several works which use fewer variations than that are known. So a simple thing would be, hey, you like a particular database, so you only select variants from that. You don't select variants from everything. You could select variants based on allelic frequency. You say, hey, I see these variations very frequently, so they must be important. I'll keep them in my graph. You could say, I'm studying a particular population, so I'm going to only limit my variance to that particular population. Or maybe I'm interested in particular loci in the genome, and I'm only going to keep stuff there. So these are all sensible, intuitively valid things. So the first work that tries to do a little bit more is Forge from Ben Langmuir's group, I think, and where they develop a mathematical model to prioritize variants. And then they select the top scoring variants according to the model. But if you look at the model, again, they're basically looking at allelic frequency, right? So giving higher priority if something shows up more frequently. And also they study the effect of keeping that variant on runtime, on space, and things like that. So these are more related to computational burden as opposed to the quality of the graph and so forth. So the question we asked is that, is there a nice mathematical framework that one could design which is meaningful and somehow defines what is the optimal number of variants that you should remove and somehow guarantee some desirable property? So while searching for that, we came to this mathematical framework where what we try to do is that we want to maximize the number of variants removed or minimize the number of variants retained such that the reduced variation graph still allows you to map any sequence of length alpha with at most delta differences. Okay, so these differences could be SNPs or indels or a combination depending on the problem. So essentially what we are asking is that, let's suppose that there is a complete variation graph, right? So that graph has every alpha long substring of every haplotype as a path. It also has many recombinant paths that you particularly don't care, and sometimes maybe you don't even want them. But those are all substrings which are found, alpha long substrings that are found as paths in the complete variation graph. Now, the reason why that graph is useful is that if you're trying to map a read, then that graph allows you to map the read. So we want to preserve the read mappability because that's really how these graphs get used. So we want to guarantee that if there is a path of length alpha that is found directly in the complete variation graph, we should be able to find the same path starting from the same coordinate with at most delta differences in the reduced variation graph. And the advantage of that is that now then you can incorporate delta into read to graph mapping algorithms. So earlier, if you tolerate a certain error rate, add delta to it to tolerate an increased error rate. And now you can do a sequence to graph mapping on the reduced variation graph, and you're guaranteed to find it. So the nice thing about this mathematical model is that you don't need to pay any attention to the biological importance of the variants that you're retaining or removing, because you're guaranteed to find the exact same thing as if you had the complete variation graph. And a nice additional benefit is that you could, if you do this only for haplotype sequences, then the number of recombinant paths would be greatly reduced. So this mathematically guarantees you very, very attractive features. So within this mathematical framework, you could ask several questions which lead to different variants of this problem. So for example, you could say, hey, I want to only allow SNPs, or I want to allow both SNPs and indels, right? So Hamming distance versus edit distance. So we realize that the edit distance case is the more practical case, obviously. But the reason for studying the Hamming distance also is that, you understand that there might be some faster algorithms for doing it and so forth. So at least you understand more about the problem and so forth. You could also ask the question about what do you want to preserve, right? So there are two types of sequences of length alpha that we consider. You could say I only care about preserving substrings of length alpha that are found in one of the input haplotypes. So if you do that, we call it haplotype aware. Or you could say, I'm going to look at every path of length alpha in the complete variation graph, and there's a sequence of length alpha corresponding to that. And I would like to preserve all of those sequences. That means that this will preserve both haplotype sequences as well as the recombinant sequences and so forth, right? So in reality, from a biology standpoint, it's haplotype aware that you need. You don't need to preserve the other sequences, okay? However, and the reason why we study that is that if you preserve all the sequences, and you will see that the algorithms are simpler and they run faster. So there is some reason to study them. You can also ask what should be the objective function, right? You could say, I would like to minimize the total number of variants retained, or you could make a rule that either I keep all the variants at a position or I remove all of them. And if you make that rule, then you say, I would like to minimize the number of positions at which variants are retained. So these three different choices will give you a problem space of 8 problems, which I'm showing you here. Basically, do you want to preserve graph sequences, or do you want to preserve haplotype sequences? And within each category, do you want to operate with SNPs only, Hamming distance, or do you want to consider SNPs and indels, edit distance, right? And then what is the objective function? Do you want to minimize the number of variant coordinates, or do you want to minimize the number of variants? So this leads to 8 different problems, which we have solved over time, and the results are cataloged in 3 different papers, which I will show you later, so that you could study those papers in more detail if you like. And you can, I'm summarizing the results here. It turns out that the first 2 problems are polynomially solvable. The rest of them are all NP-hard. The very first problem can be solved in linear time using a greedy algorithm. The second one can be solved using linear programming. And then from then on, they are all NP-hard problems, and you could solve them using various types of ILPs. And the complexity of the ILPs grows as we go from left to right in this diagram. So that's a snapshot of how our results pan. Okay. So there are NP-hard proofs for the last 6 problems. There are algorithms for all the 8 problems, and I'm looking at maybe 5 to 10 minutes. So I'm just going to give you a sample of a couple of these ideas, and then I will show you some results, and then leave the papers for further study if you're interested. Okay. So the greedy algorithm, for the case where we want to retain all strings of length alpha found in the complete variation graph, is very simple. So we are considering SNPs only. So your variation graphs looks like this. The greedy algorithm basically says, hey, let's position, right, a string of length alpha like this. Okay. So for a variant in position I, this variant will be included in all alpha long paths that begin anywhere between I minus alpha plus 1 and I, right? So if you have you can represent this by a line, and as this line moves from left to right, this position is included in this line starting from this being the origin of the line as you keep moving the line. But once you cross this and go to the next node, then I will drop off essentially. So this variant will contribute to an error if you remove it to all of these alpha long substrings. So all you need to do is that place it in the first position, and then ask the question, how many of these variants are here? And you are willing to tolerate at most 2 errors. So you essentially remove the first 2. And then you say, okay, when I came to the third one, I already have an error rate of 2. I can't tolerate anymore. So let's retain this variant, because if you retain a variant, then you won't have an error in mapping. And if you remove the variant, then you will have errors in the mapping. It's that simple. Okay. So it's a greedy approach where you keep throwing things away, and then once you hit the limit delta, then you start retaining them. But as the line moves, some variants go out of the window. So the previously removed ones are no longer a problem, so you can remove new ones and so forth. So that's how essentially this works. And you can prove that this is optimal. This just takes linear time. because you don't need to even consider every position. It's linear in the number of variant positions, because for every variant, there is a window, there is a starting point where it starts to influence the errors, and there is an end point. So there are two events, and you can do a line sweep as in computational geometry, and you can solve this problem in linear time. And you can show that greedy gives you an optimal solution, because if not, assume that you have a greedy solution given by G1 through GP and an optimal solution that is given by O1 through OQ. Now pick the first instance where these two solutions differ, and let's say that is at the index K. Up until then, the GIs and OIs are the same, but for the first K, GK is not equal to OK for some position K. Well, the greedy algorithm postpones retaining variant as much as possible. So the optimal one has to retain a variant before that. So if that's the case, you can simply replace OK with GK, and the solution will still retain optimality, because the influence of retaining OK will end before the influence of retaining GK. So the greedy algorithm is actually making a better choice. So it's possible there is another optimal solution, but you can convert that to, you can use that to prove that greedy is optimal as well. So the nice thing about the linear time algorithm, this is not even linear in the size of the graph, it's linear in the number of variant coordinates, is that it runs extremely fast. And one important thing to notice is that when you are building these variation graphs for these 8 problems, right, in practice, you could use any of these variation graphs, right? So basically, as you go from left to right, you're essentially achieving more and more reduction, right? So if you only care about preserving haplotype sequences versus graph sequences, then you can remove even more variants and so forth. But to hunt for them takes more time. So if you don't do this, the other graphs will still be useful to you. So any graph can be used, but the graph sizes basically decrease as you move through this problem space. Okay, so that's a nice thing about this.
Srinivas?
Yeah.
That's Pierre. I have a naive question. I didn't understand the difference between the variant coordinates problem and the variant problems in your sub-problem tree.
Oh, okay. Okay. So the difference between variant coordinates and variants is that if you want to minimize the number of coordinates, that is the number of positions at which you would like to remove the variants, right? You want to maximize the removal of variants or minimize retaining of the variants. So you have a rule that at every position you want to remove everything, right? If you relax the rule and say, hey, I'm okay to remove a subset of the variants, right? Then you can think of a mechanism where you would actually minimize the total number of variants removed, right? So that is that's the difference between the two objectives. Is that okay?
Okay, thank you.
Okay.
So we have another question from Ren.
Sure.
Sorry. So my question is, to what extent removing some variants is biologically correct? I mean, removing some variant may affect the biological aspect of the variation graph at the end.
Okay. Yeah. So here I'm confident to say that you really don't need to look at biological relevance whatsoever at all, and you'll be completely safe. And the reason is that the mathematics is guaranteeing you that any read that you can map in the complete variation graph, you will be able to map the same read to the same position in the graph, and from there the same position in the haplotype if you like, provided you increase the error threshold that you tolerate of your sequence to graph mapping algorithm by the chosen delta, right? So the mathematics guarantees that you will find it. So there is no need to worry about anything else. Okay, so the mathematics may even suggest that you throw away something that is extremely considered important from a biology point of view, and it does not affect the outcome because you'll still map to the same location. And once you know the mapping, then you know where it maps in each of these haplotypes, and then you can recover that biologically meaningful variation, basically. So even if it's not in the graph, you will be okay because you can find it. Okay.
Thank you.
Yeah. Any other question?
I have one. So, if you're using R1 as a backbone and R2 and R3 are just getting added, when you're minimizing variant count, you're going to remove the variants from R2 and R3, right?
Yes, but if you like, you can keep any of the variants that you like, and it will be the same number, and that will be completely all right.
Okay, thanks.
Okay. Okay. So,
sorry.
Yeah, go
Finally,
ahead.
I have another question. Sorry. Maybe it's naive, but how can you be sure that you are not going to change the mapping position of a read if you remove some differences that affected the original position of this read on the graph?
So when you run when you run a graph mapper software, right? I mean, any other software, it doesn't need to be ours or anything, right? Take any sequenced graph mapping software that anybody wrote, right? It will basically ask you what is the edit distance threshold, right? When you're doing a mapping, you have a certain edit distance threshold. So that threshold you choose is a function of the errors that you expect the next generation sequencing machines to make, maybe the genetic differences that you expect between the individual and another individual of the population. So trying to take this into account, you allocate an error rate, right? So what you need to do is that increase the error rate that you tolerate by this delta. Okay, so you pick a value of delta to come up with the reduced variation graph, and you increase your error threshold that you tolerate in the sequenced graph mapping algorithm by the same delta. That will guarantee that you will find everything.
Okay, but not necessarily at the exact same position as the original.
No, it will find at the exact same position.
Okay. Okay, I will try to understand this later, maybe.
Yeah, you will not miss anything. Right? It's a promise backed by mathematics, so it's correct.
Nice, still.
All right. So I will go through this somewhat quicker here.
Hello?
Yeah.
Hello? Connecting with the question of pair. Sorry, it's an echo here. Oops. I can still.
Okay, I'm hearing an echo.
I can still see that. Okay, you still get the best alignment. So in case of unique alignments, you get that one. But if you have multi alignments and they're mapped to multiple places, then you might lose a couple, right?
No, you will not.
That is true.
You will not.
Isn't that true?
No, you will not lose.
So if you have ambiguous alignments, yeah, you preserve that you get it mapped from to where it comes from in a way, yeah. But on the other hand,
no. So
like,
the reason Alex is
slightly modified other places where it could map to, and this could be important for you, then you miss this possibly, right?
No, you'll not miss it. The reason you'll not miss is that if the sequenced graph mapper, when applied on the complete variation graph, answers you saying that there are these three different positions where it maps, you will find the exact same three positions here, because the same sequence, the paths are preserved subject to losing at most delta differences. So if you increase the tolerance by delta, then you will capture every one of them.
Yeah, but maybe two of these positions could already be affected by mutations, and then if you get rid of a couple more, then you exceed the threshold of delta.
No, but you're adjusting the threshold to account for it, right? So if you're running the sequenced graph mapper on the complete variation graph with an error threshold of delta 1, let's say, right? Then when you use the reduced variation graph according to our technique, you change the threshold from delta 1 to delta 1 plus delta.
So this is the trick, and then you get this error.
And then you're guaranteed to find exactly
that I agree on. Yeah. Okay, sure.
Right.
That's fine. Yeah. Okay, thank you.
Okay. All right, I'm going to go back to the presentation mode. Unfortunately, when I go into that mode, I won't be able to see if your hand is up. So please, please speak up if you have a question. Okay. All right. So the rest of the problems and the algorithms for them are essentially using integer linear program formulations. So let me show you a very quick one. So this is again the easy case of SNPs only, no insertions and deletions yet. Okay, and we are trying to minimize the total number of variants. So the way you formulate this problem is by creating a cost function here. So what we are doing here is that suppose you have n variant coordinates. So you have these two vectors, c and x. x1, x2, x3, x4 are basically variables which take on a value of 0 or a 1. If it is, it essentially tells you whether you are keeping the variants or throwing them away. Okay. And then the C would be the vector that tells you the cost benefit of throwing them away. So for example, in this position, there are 4 SNPs. So if you throw away SNPs at this position, you will have a benefit of 3 because you have to at least keep 1 to keep the graph connected. So the benefit here is 3. So it's really 1 minus the out degree. So that's the cost that you see. And you're trying to maximize C transpose times X. Okay, so that is what you're doing, subject to a set of constraints. And the constraints are basically what we call these alpha delta compatibility constraints. So if you place a path of length alpha, you want to tolerate no more than delta differences, right? So therefore, essentially, you have the same x1, x2, x3, x4 here. And if you throw something away that contributes 1 to the difference, and so you want to make sure that this matrix vector product is less than or equal to delta. So this is how you formulate the problem. Now, in this particular case of SNPs only, it turns out that this matrix has a very interesting property. It's a 0-1 matrix, and it also has ones consecutive in a particular interval. So this property makes sure that if you solve this problem using linear programming technique, using LP relaxation, the answer you get is guaranteed to be an integer, and therefore you don't need to use integer linear programming, and you can use the regular linear programming, and that's what makes this particular problem polynomial. It's a lot to take in, but I will make just one more observation, which is that if you don't care whether the sequences are actually found in haplotypes or not, and you want to preserve all the sequences found in the graph, then you essentially have to assign these variables to positions, right? So x1 for this position, x2 for this position, and so forth. Okay. The reason is that whether you find it in a haplotype or not, you can take an arc here, and then you can go and you can take another arc here, and so forth. But if you have a haplotype aware version of the problem, then you have to put these variables on the edges, because there are rules that if you go over this edge, then you must go over only a certain edge here, because the other edges along with this edge are not found in the same haplotype. So the number of variables will increase by many-fold if you go from graph aware to graph path aware to haplotype aware version of the problem. And then the ILPs, it's an NP-hard problem, so the ILPs become very, very difficult to solve. Okay, so that's really the general idea. So let me quickly jump to some results, right? So that, so there are really NP hardness results, and there are also multiple algorithms for every one of these problems that are cataloged in detail in the papers. And I don't want to spend too much time here, but you can see that this is really the simplest problem here, the problem number 1, and that itself can remove so much, okay? So the runtime here, as you can see, is extremely small for greedy algorithm, because it's a linear time algorithm and linear only in the number of variants, right? So we are done extremely fast here in terms of time, right? I'm sorry, I forget whether this is in minutes or seconds, but in any case, it's really, really small. And you can see the reduction that is achieved. So as you increase delta, you can achieve a higher and higher reduction. So that's exactly what you're seeing here. The same thing is cataloged in terms of bar charts here, where if delta is equal to 0, which means that you're not allowing removal of anything, so that shows like all of the variants. But as you increase delta, you're able to reduce the number of variants retained. So this is not really drawn to scale because we couldn't really capture the extent, but this is really 6 million here, and this is just 13,650. So that's really a great reduction. Okay, and so this is at delta equals 5%, you lose about half of them, and so forth. So this is just for the problem version 1. I'm going to skip a few more algorithms to save time here. Let me show you the result for haplotype aware version of the problem. So this is really problem number 8, so which is the rightmost problem, where you only try to retain sequences that are found in haplotypes, which is actually what is relevant from a biology standpoint of view. And you want to incorporate both SNPs and indels, because you need to take care of both of them. So this is the biologically most meaningful thing. And the reason why we got to it after a couple of years is because this will require modeling too many variables, and it leads to very large ILPs in an NP-hard problem. So the way we were able to crack it was to recognize that if you have a long stretch, longer than alpha plus delta, where there is no variant, so you will find these regions where there are no variants, they form a natural breaking point, so that instead of having a one gigantic ILP, you can break it into a large number of sub-ILPs, and then these can be solved independently, so you can use multiple cores and so forth, and so you can put some heavy-duty computing, multiple resources, and crack this problem. So you can see that this really achieves the greatest variant reduction. Just focus on this column that shows percentage of variants removed. So you are seeing that even for something very small like delta equals 1, you can remove almost 90% of the variants. And if you're willing to go up to delta equals 2, you can remove like 97% of the variants. So the variant reduction that we achieve is extremely significant. So the variation graphs become very, very small after you throw away so much, and yet they retain the attractive property that you will be able to map as before. Okay. The results that I presented are chronicled in 3 of these papers. The first one is a 2021 ISMB paper that essentially solved problems 1, 2, 3, 4. And then the follow-up paper in 2022 in ACM-BCB that solved problems 5 and 6. And we just submitted a paper to ISMB again that solves problems 7 and 8. I'm happy to send you a preprint of the last one. If you like, we have no clue, of course, whether it's accepted or not. So I'm happy to share with you if you like, but these papers will essentially walk you through all the NP hardness proofs and all the algorithms. And we also wrote software, so you don't really have to, follow these algorithms, except if you want to be, convinced that this is a good approach. But you could click on these links and you can download the software, and then you could use any of this to achieve the variant reduction. And once you let our algorithm and software to select the reduced set of variants, then you can feed it into VG Toolkit and things like that, and say, hey, build me a variation graph or a genome graph for these particular variants, and it will do that. So you can use that in conjunction with that. So this marks the end of the first part of my talk. I'll stop here if there's a quick question or two before moving to the second part. Okay, Alex?
Yes, okay, so there will be echo again. Mm-hmm. So thank you very much for the lovely first part of your talks. I was wondering whether, very often you start searching for the right location of your reads in the graph or in the sequences by running a k-mer-based index or something like this kind of, you're looking for seeds. So
yes.
All I understood is that your approach also really nicely caters to filtering-based approaches, or are there any turns that keep me from filtering?
Well, that's an excellent observation. Yeah. Something that I should have mentioned in the talk is that you get to decide what alpha should be. So if you have a sequence to graph mapping algorithm that uses seeds and then seed and extend approach, then whatever seed length you choose, that should be alpha.
No. Sorry. I forgot to put my loudspeakers on. I'm sorry. I haven't run anything.
Oh, okay,
Hasn't done
okay.
anything.
No,
Okay,
that's
one
all
more
right.
time.
So,
Thank you. It's a math here.
so I was commenting that that's an excellent question. And what you need to do is that if you use a seed and extend algorithm, then you have in mind a certain length for the seed, and you choose alpha to be the same length. Okay, and then the graph will give you the property that you're looking for.
Yeah, I was thinking if you divide alpha by, so you have like you have given alpha and given delta, then you can be sure that there's a minimum length that will be matched exactly, yeah, for each of the reads that you draw from the haplotypes of the graph sequences.
No. I think a better way to think about it would be that you have a sequence to graph mapping algorithm.
Yeah.
And when you run that algorithm, you either take the entire read and say, hey, go find this in the graph, or more likely you take you break the read and say, hey, I'm only looking for a seed, because mapping the entire read may be hard and the algorithm may not do that. Particularly for long reads, you may not want to go, map 10,000 bases, right? You may say, I want to pick a seed length. Now,
Yes.
within that seed length also, you may want to tolerate errors, right? Because if it's a long read with 10% errors, even if you take a long seed, it may still have errors and so forth. So whatever is the length of the sequence, whether it be the full read or the seed that you would like to map, using the sequence to graph mapping algorithm, you choose that length to be alpha and do variant reduction, then they'll work together very well.
Yeah, of course. Yes, this is an easy solution to my yes. Okay.
And like if you are interested in using it, and if you come up with some questions, or if you just want to verify whether you're using it correctly, feel free to send us a mail, and we'll be happy to go over that with you.
Yeah, but you have to for the particular application, for example, for seeding, you have to rerun the algorithm and remove it accordingly, remove the variant positions of the variants accordingly. And also for if you would like to crank up the number of multi-read alignments, then of course you also need to
so you do have one.
So you have to modify.
That helps you, right? If you tolerate at most delta errors in a window of length alpha, you're also tolerating at most delta errors in any smaller length. So therefore, you can use the variant graph for a smaller length mapping also, smaller than alpha. The flip side is that you may be unnecessarily spending more time, right? That's the only thing. But other than that, in terms of accuracy of the results, if you build it for alpha, then you can use any length less than alpha.
Okay. Sure.
Solon, you have a question?
Thank you. Yes, very fast, Srinivas. So I saw that also for the harder problems, I mean, formally the NPA hard ones, you also had a word greedy. Is it, I mean, this greedy, is it a heuristic or is some approximation? I mean, what is, because you had greedy / ILP, what is this?
Right. Yes. So for the first problem, greedy gives you the
yes, this is optimal. Yes,
right?
This is optimal. Yes.
Yes. For the other problems, you can use greedy as a heuristic. It doesn't give you.
It's heuristic.
Yeah.
Okay. This was my question. Okay, thanks.
Yes. So heuristic would remove fewer variants than it could have, but remember that the variation graphs are always useful, right? So, okay, fine, you could have thrown away more, but you did not, which means that your graph is a little bigger, but it is still useful because it will still find you all the mapping positions. So from a biological point of view, if you don't like to spend time optimizing the right problems, you can take the left side of the tree and you'll be fine.
Yes. Okay? Clear. Yeah.
Okay, all right. Okay, so it seems like I have only maybe about 10 minutes left in the hour. So I appreciate your generosity in sticking around a little bit more, but I will try to race through this a little faster so that I don't spend 40 minutes going over this. So this is the second part of the talk where I want to talk about aligning sequences to graphs. And here we have done both theoretical work as well as practical work. Hank described one of the practical papers that we have, the GWFA, in the last session that you had. So I want to show you some quick results here. First, let me start, presenting the theoretical results. So there are lots of sequence to graph mappers, but the complexity of the problem is not settled, right? So is it a problem that admits an efficient algorithm? Is it NP hard? This question is not settled. So the first thing we wanted to do was to first understand this problem. So we are modeling a sequence graph. This could be variation graph or any other type of graph, where this is basically a graph that consists of sequences. And now we are modeling strings to be within the nodes, right? So there are many ways of representing these graphs. You could have characters, individual characters only, or strings. You can have them at nodes or as edge labels and so forth. And many of them turn out to be equivalent. You can transform one from the other. So in the different papers, we may have started with different representations simply because of the choice we made at that time, because these papers are written at different points of time and so forth. But the results would easily translate, and you can change it from one graph to the other. So sequence to graph mapping, again, all of you know this problem, so I can go through this very quickly, the intro slides. The one key thing I want to note is that when you're trying to do a sequence to sequence mapping, right, then we have all of these things like, hey, what should be the scoring model, right? Hamming distance, edit distance, affine gap penalty, linear gap penalty, and all of those things, right? And you have the type of alignment. Is it global, semi-global, local, and so forth, right? So you have all of those complexities when you do sequence to graph mapping. In addition, you have three other things to think about, right? When you are doing the sequence to graph alignment, do you allow changes in the sequence? Do you allow changes in the graph? Or do you allow changes in both? Okay, so basically that magnifies the set of problems that you have. And the reason why changing in a sequence versus changing in graph is different is because when you are doing a sequence to sequence alignment, whether you make changes in the first sequence or the second sequence, the number of differences is the same, right? You can change the G's in the first sequence to C's and it'll agree, or you can change the C's in the second sequence to G's and they would agree. With respect to a sequence to graph, what happens is that if you make changes in the sequence, then you have to make two changes. The two G's have to become two C's. But if in the graph, you can just make one change the C to a G, and then this would map the sequence, because you're going through this in a cycle to do the mapping. So if you have a cyclic graph that also allows cycles, then you may have fewer changes in the graph that will allow you to make the two sequences equal, right? So whether you have changes in sequence or graph, whether you allow them in both, can change the nature of the problem. So an interesting thing about this problem is that, back in 2000, Amir studied the same problem in the context of mapping a string to hypertext. And he has proofs there showing that this problem is NP complete if you allow changes in the graph alone, or if you allow changes in both graph and query. And the problem where you only allow changes in the query is polynomially solvable. So he has this classic paper a long time ago, and we can just make that paper applicable to computational biology. But there is only one problem. Amihud's proof assumes that the alphabet is larger than the size of the graph, okay, in order to do that proof. But here we are trying to build graphs for DNA sequences, maybe protein sequences. So the alphabet size is very small, like 4 perhaps. And in any case, it is a finite size. So his NP completeness proofs don't apply. So the first question we wanted to settle and understand is that, is the problem going to be NP complete even for a small constant sized alphabet, or are we going to get lucky somehow that this problem is going to be polynomially solvable in the case of computational biology? If that's the case, that would be really a great result, right? So we wanted to study that first. And so what we ended up showing is that the problem remains NP complete for any alphabet of size greater than or equal to 2. Of course, alphabet of size of 1 doesn't even make sense because there's nothing allowed, right? It captures nothing, right? So in that sense, this problem will remain hard. And we also came up with an efficient algorithm for the case where the changes are allowed only in the query and not on the graph. And we can also prove that this is a conditional lower bound based on the strong exponential hypothesis that you cannot really beat and get a faster runtime than this algorithm. So these results are canonical in a recomp paper here, and you can read this for more detail. So I wanted to show you these proofs, but I'll go through them very quickly in the interest of time. So essentially, to show that this problem is NP complete, even for alphabet size equal 2, what we do is that we take the directed Hamiltonian cycle problem, the problem of finding a Hamiltonian cycle, a cycle that walks through every node in the graph and comes back to the same position in a directed graph. So that's a known NP-hard problem. So we solve that by keeping the graph the same and putting string labels in the vertices of the graph. So in this case, what we need to do is, if alpha and beta are your two characters, we put alpha to the n in every node in the graph. And then we have a query sequence that corrupts these alphas by introducing a beta strategically at different positions and so forth. And the reason for doing this is that we want to force that if there is a Hamiltonian cycle in this problem, then this query will map to this graph, to this sequence graph, making no more than n errors, where n is the number of vertices in this graph. So you have these proofs. And then if you can map the query, then you found the solution to the directed Hamiltonian cycle problem. And since that problem is NP-hard, this will be NP-hard as well. So you can prove this for Hamming distance. It's a little bit more complicated for edit distance. So we have 4 proofs, one for Hamming distance, one for edit distance, and then one each for these 2 problems, whether we allow changes in only the graph or you could have changes both in the graph and query. So all of this turns out to be NP-hard. Okay, so that means that your sequence to graph mapping algorithms that operate in this space can only be heuristic or approximate. But if you allow changes in the query alone and you treat the graph as sacrosanct, then you can actually achieve an optimal runtime. So essentially, that is the other result. So the algorithm very quickly is that instead of using dynamic programming tables, we use this concept of an alignment graph, where if you're trying to align a query, in this case ACT, to an alignment to a sequence graph, which is in this case like this, and so as you know, we do admit cycles, and that's not a problem here. So this solution will work even for cyclic graphs. So basically, think about making as many copies of the graph as you have characters in the query, okay? And then you have 3 types of edges, essentially. The red edges are insertion edges, which means that they are the same edges as you see in the sequence graph, no different. They operate within a layer. And this means that you're jumping from one to the other without consuming any character in the query. So that makes it an insertion edge, right? Or you have vertical blue edges, which are deletion edges, which means that you're advancing on the query string, but you're not advancing in the graph. Therefore, it's a deletion. And then you take these same edges and you go from one layer to another layer. So if there is an edge from A to C, you'll go from A in one layer to C in another layer, the green arrows. These are the substitution edges. So there may be matches or mismatches. And if it's a mismatch, you would have a penalty. If it is a match, then you don't have a penalty. And then you can compute edit distance. And you can very easily show that the alignment, a valid alignment, really is captured by a source to sink path in this alignment graph. So we use that model, and we do a couple of nice algorithmic tricks to try to find the answer. You can do Dijkstra on this because it's a graph and you're trying to find the shortest path. But if you do that, it will take this much time. But this alignment graph has very special properties because we're trying to do edit distance based alignment. So all the green edges and all the blue edges and all the red edges have the same cost. So because of that, you can do away with priority queues and you can maintain things in sorted order. You can prove that things do not violate the sorted order as you go from one layer to another layer. So the way we do this is that we compute the shortest path lengths from the source to every layer, one layer at a time, because there are no back edges here. So let's say that you have already computed things for layer 1, and then you want to compute things for layer 2, okay? So then we have 2 stages. In the first stage, we only look at the green edges and the blue edges. saying that, hey, if I know the optimal way of reaching every one of these vertices in layer 1, how can I optimally reach every one of these vertices in layer 2? It's very simple. You look at all your incoming edges, you pay the appropriate penalties, and you see what minimizes the distance, and you take that. So this part is linear, obviously, because you have edges coming in, and you can just look at it, and then whichever gives you a better edit distance, you take it. So you can do it that way. The only problem with that is that you're not taking care of internal red edges, right? And therefore, you are going to miss these insertions, right? Maybe there is a way to insert some, take some insertion penalty and then get a shorter edit distance. So you can propagate that insertions and maybe come up with a reduced cost. And we have an algorithm to do that without incurring log penalties and not maintaining priority queues. And that is because the cost of all of these red edges is the same. Whatever that cost may be, it doesn't have to be unit, it can be any number, but it's a fixed cost. And therefore, if you maintain things in a sorted order, and then those distances change by a fixed amount only, then you can maintain multiple sorted orders for different distances, and you can merge them and do some standard computer science tricks. And so we do this to come up with optimal algorithm in this particular case. Okay. So anyway, so this is really the final result that we have, that we can we have the optimal algorithm when changes are allowed only in the query and not in the graph, that would work irrespective of whether you choose timing distance, edit distance, linear gap penalty, or affine gap penalty. In all of these cases, you get this optimal runtime, and this is optimal under the strong exponential hypothesis assumption. And then the problems are NP complete if you allow changes in the graph at all, whether it is graph alone or in conjunction with the query. So in the interest of time, I'll stop here, but I'll refer you to a bunch of papers. So these are papers that give you the algorithms and theory. And in the top two papers are basically recomp papers, but I refer you to the Journal of Computational Biology extended version because it has more details. The first one shows that sequence to graph alignment is NP complete, even for finite sized alphabets. It also gives that efficient algorithm that I showed you. Then the next question was that, hey, what if we restrict the graph to graphs that are frequently used in computational biology? Will that become polynomially solvable? So my postdoc, Daniel Gibney, who is attending this talk as well, was able to solve this problem, even for De Bruijn graphs. It's a very complicated proof that only he could do, actually, in our group. But the net result is basically that the sequence to graph alignment remains NP hard, even if you restrict the sequence graph to be just a De Bruijn graph. One other paper that I haven't talked about here that I would refer to is that mostly, in fact, as far as I know, all the sequence to graph alignment algorithms talk about mapping one sequence. But when you sequence using Illumina and so forth, right, you have these paired reads. And so it would be nice if there is a way to validate these paired and distance constraints to ask the question, hey, I'm mapping both the reads of the paired end. Is there a path in the graph that conforms to this paired end distance? Otherwise, there is something wrong with the mapping, right? So we have a paper that shows how to validate these paired end constraints after you do the mapping. And we also have some practical papers. The paper with Heng Li, Heng presented last time. It's a fast sequence to graph alignment, which is sensitive to edit distance. So if the edit distance is small, it'll accelerate the alignment by multiple orders of magnitude. We also have another paper which is related to mapping multiple sequences in a shared memory machine in parallel. Because when you try to do sequence to graph mapping, you rarely do only one sequence and stop, right? You have a bunch of reads coming from an NGS sequencer. You want to map all of them. So rather than map them one by one, if you map them collectively, you can take advantage of the shared memory parallelism there. So we have a paper that showcases this as well. And I'll be happy to send you a PDF copy of these slides so you have access to these papers. And if you like, you can read more about this work. And if you have any questions, please feel free to just email me, and either I or one of our group members will answer. And one last thing before I conclude is that Pierre mentioned that I direct the Institute for Data Engineering and Science at Georgia Tech. It's a pretty large institute. We have 200 plus affiliate members. We are located in this very nice 21-story building on the 12th floor. We have visitors' offices and so forth. So if anyone in your group is interested in spending some time with us, working with me or my group or other faculty members at Tech on any of these problems or other things, please let me know. And for faculty there, if any of you want to do sabbatical, we can do that hosting as well. So I'm taking this opportunity to invite you all. And if it works out anytime in the future, please consider it an open invitation and feel free to contact me. I'll stop here and then take any questions. And I have as much time as you like, but I don't want to tax you, so please feel free to stay and ask questions or leave or whatever you like.
Thank you. I think Solon has a question.
Yes, thank you, Pierre, and thank you, Srinivas, for this wonderful talk. Just a very small comment for the second part from me is that even the M times E lower bound, just to clarify that it's a lower bound also for no distance at all, for just pattern matching. And
Yes.
This was shown the same year with you, but in iCalp by some other authors. Yes. It's just to say that also other authors have shown this. So even with no edit distance or coming distance, just pattern matching is quadratic. Just to say that.
Yes, thank you for bringing that up. That's an excellent observation. So we are showing an algorithm which meets that lower bound that can take edit distance into account, but the lower bound itself is valid even if you don't tolerate any errors at all and you want to do exact pattern matching. So the algorithm cannot improve beyond this.
Yes.
But one of the things that I would mention though is that in our own implementations, what we found is that sometimes the Dijkstra's algorithm, even though it has a worse runtime complexity, actually does better in practice. So one has to take the theory always with a pinch of salt, right? So one has to implement, but theoretically this would be the optimal algorithm. It's actually runs quite well, but there is always some overhead in maintaining these multiple arrays and so forth. And one of the nice things about Dijkstra's algorithm is that if you try to go from the source to sink, you can do it without going through every layer one at a time. So if there is a short edit distance, right, you can quickly find that path. And so that's where it still wins in practice.
Yes, thank you very much. Just wanted to clarify this. Yes.
Okay, I think someone else has raised his hand, but I cannot see who it is.
Okay, so
Only one. Okay. Is there any other question? Personally, I have one, but it's related to the first part of your talk. I don't know if everybody get bored or not, but I have a question. Have you thought about the not selecting variants after the graph creation, but selecting variant at a graph creation when you had some genomes dynamically?
Yes. So the complete variation graph is basically an abstract concept. You really don't have to build it. So we essentially start with SAM tools and so forth, right? What we require as input is typically you have one haplotype, and then the variants are described with respect to the haplotype, right? So all of our software takes that as the input. So the complete variation graph is essentially an abstract concept in mind to compare what we are doing and to explain the algorithm, but you don't actually have to build it or use it.
Okay, thank you. Okay, so no question here. I think we can thank you again. Thanks. That's very nice.
Thank you so much for giving the opportunity. I have read about your projects when they were launched, and then we do try to keep up with the papers as much as we can. So really appreciate this.
Thank you very much.
Thank you very much. It was very interesting.
Thank you.
Thank you. Bye.